Eventually somebody's going to forget and everything is going to crash. Okay, it's 1 o'clock. We got 48 people, which is a pretty healthy number. It's still increasing. But I think it's probably time to get started. I want to welcome everybody to the doctoral defense of Alyssa Liguori, who's going to regale us with her research for the next hour or so. I am the chair of the dissertation committee. I am Stephen Baines, and I'm going to introduce the rest of the committee now and sort of go over some ground rules. First, we have visiting us from USC, from University of Southern California, Suzanne Edmonds. She is, we thank her for participating in this committee. She's been very useful. We also have Joshua Rest, and we have Bob Thacker, who is acting as Alyssa's advisor for this dissertation. Now, the ground rules are very simple. We, the committee, do not ask questions during this public, at the end of this public defense. Alyssa will give a public presentation of her work. At the end, she'll open up to questions for from the audience, but the committee will not actually be asking you those questions. We will retreat to grill her later in private about the details of her dissertation, and at some point, hopefully, we will come out all happy. Alyssa's got some very specific instructions to the people listening to her defense today. First, we want to make sure you turn off the video, your video, until the question and answer session. That's to avoid distraction and reduce bandwidth issues. If you want to ask a question at the end of the presentation, you want to click on the participants option and use the raise hand button. And that way the other hosts will be able, and Alyssa and the other hosts will be able to pick on you and unmute you so you can ask your question. If you're having any technical issues listening to, hearing or seeing the presentation or getting access, please contact Allison Rugula, Dave Charison, or Jose Nunez, who are basically doing the background hosting for this presentation. Okay, so without any ado, I'm going to give the microphone over to Bob Thacker, who's going to introduce Alyssa.
Great, thank you, Stephen. Hi, everybody. It's my pleasure to introduce Alyssa Liguori today. And one thing I noticed about Alyssa is that she became interested in invertebrate biology when she was an undergrad at Dowling College, also here on Long Island. But she has 2 co-authored publications on crustaceans of Easter Island from that undergrad experience that was really formative for her. Another big experience that she had here was that her first summer in our program, she went to the University of Washington's Friday Harbor Laboratories, and she discovered that that's an amazing place to work. So she continued to return there for the next 3 summers. She received a National Science Foundation Graduate Research Fellowship to support her research, and then numerous other grants and scholarships from Stony Brook and Friday Harbor and the American Museum of Natural History and several other institutions. So Alyssa's been intrigued by this ability of animals to respond to changing environmental conditions, particularly through both phenotypic plasticity and adaptation. But she's also interested in this idea about how maternal environments can impact the offspring. So at Friday Harbor, she was able to use their state-of-the-art ocean acidification lab to conduct some pretty complicated experiments where she altered pH, temperature, and salinity. And she also found a great study organism there that she's going to share with us today, this copepod, Tigriopus. But in addition to all those experiments, Alyssa found that she needed to add some molecular and bioinformatics skills. So she obtained an NSF graduate research internship at the NOAA Northwest Fisheries Sciences Center. And then she applied those techniques to her system, and she's going to share that with us today as well. So in addition to the research that Alyssa is going to share with us today, she's also been involved in several other service activities while at Stony Brook. She's had leadership roles in the Graduate Women in Science and Engineering program, GWISE, and she's mentored several undergrads to that program. And at a national level, she served on the executive committee of the American Microscopical Society as their graduate student representative and led the social media efforts for that society. So Alyssa's been made some pretty big contributions to us in the department and on campus, and then as national service as well. So today she's going to share with us an overview of her dissertation, and the talk title is, or the dissertation title is Population Dynamics in the Rocky Intertidal Zone, Acclimation and Adaptation to Extreme Abiotic Conditions in the copepod Tigriopus californicus. So I'll now turn the microphone over to Alyssa.
All right, thank you so much, Bob. I hope everyone can hear me and see my slides. All right, so here's my title slide. So thank you so much for that introduction, and thank you everyone for joining me virtually to hear about all of the work I've been doing both at Stony Brook and at Friday Harbor Labs and the NOAA lab in Seattle over the past, I guess 4 years I've been doing this research. And so just here's my talk overview so you know what to expect. First, I'll give a broad introduction to my research topic, and then I'm going to tell you a story in 3 parts. So the first is where I explore multi-generational responses to pH in distinct populations of Tigriopus. In part 2, I'm looking at population differentiation using morphological traits and some genomics methods. And then in part 3, I'm exploring local adaptation to salinity and temperature treatments, as well as their variability using combined field and laboratory studies. And then I'll end with some more kind of broad conclusions. And so to begin with, kind of the very broad question that's driving my whole dissertation is how do organisms endure shifting abiotic challenges across scales in both space and time? And so this is, I think a question ecologists have been asking forever, but it's becoming more urgent recently as human activities are influencing local environments in many ways, including with climate change. In marine habitats, there's ocean acidification, pollutants, you name it, we're changing habitats worldwide. And so there are different mechanisms that organisms at different scales of biological organization can employ to endure these shifting abiotic challenges, both changes in environments in space as well as over time. And so what populations can do is track suitable habitat if it's available. So if where their suitable habitat is shifting due to something like climate change, maybe they can follow that shift along either latitudinal or elevational gradients. And this has been found in a range of species. And this study is an example where they looked at small mammal species in Yosemite National Park and found that some high elevation species had their lower range limits contracted, and then low elevation species kind of expanded their ranges upward on mountains to be able to track their optimal, thermal niches, which are changing due to, and where those places are is changing due to climate change. And what can also happen is that individuals can acclimate, or we also call this phenotypic plasticity, which is when one genotype, an individual with set genes, can produce many different phenotypes or traits with that same genotype. And so this can occur within individuals, within generations, and this can be reversible. And I'm sure you can think about many ways in which your traits can change over time in response to environmental conditions, say maybe as the seasons change, and then when those traits can kind of keep going back and forth. But there's also irreversible plasticity that can occur, which is sometimes called developmental plasticity. So that's when, environmental conditions that you experience early on in life kind of shifts the traits that you express, and those are permanent changes for the rest of your life. But plasticity can also occur across generations, and this is given many names, sometimes maternal effects or in general parental effects, because fathers can have transgenerational effects on their offspring as well. This is generally called transgenerational plasticity, meaning, the environmental experience of the parents is then influencing the offspring and their performance, but this is not changing the genetics. And so an example of this is that in the American bellflower, the maternal light environment can influence the offspring life history and whether they are annual plants or biennial. And this has been shown to be adaptive, and if the offspring's light environment matches the mother's environment. And so this is just one of many examples in which the experience of parents can influence how the offspring perform and what their fitness is, again, without any change in genetics. And finally, on the population level, there can be adaptation, right? So this requires genetic variation within the population upon which natural selection can act. And one of my favorite examples of this is work done by Alyssa and colleagues, where they were studying purple sea urchins all along the west coast of North America in the California current ecosystem. And they actually found that, currently these purple urchin populations are locally adapted to these mosaic pH conditions that occur in this habitat. And this is due to upwelling. So some parts of the coast have very low pH and some parts of the coast have higher pH. And in addition to this local adaptation, they found that you could take high pH adapted populations and then select them in the lab in low pH conditions. And you can see that similar genes are being selected in the lab versus genes that have been selected in nature in the field. So this is one of, again, tons of examples that are becoming more abundant as people try to research how populations may be able to adapt to global change. And so I've shown you many examples of different taxa and environments, but the environment that I'm going to focus on today is the rocky intertidal zone on the west coast of North America. And I've chosen this as a study system because it's a very interesting but extremely harsh environment. Some people say it's one of maybe the most harsh environments on the planet. And this is because it's where marine and terrestrial ecosystems meet. And in the span of a few meters, as you can see here, the organisms that persist here might have to face stark gradients in many abiotic factors, including temperature, salinity, pH, physical disturbance, desiccation, stress. So here's one of my favorite field sites, Cattle Point, which is a prime example of the rocky intertidal zone. And so just to show you some abiotic data associated with these kinds of habitats, this is data from Morris and Taylor, and they studied tide pools that occur kind of lower on the shore and then a rock shore. And here we're showing you daily trends in temperature, dissolved oxygen, and pH. And what I want you to focus on here is that, there's wide daily fluctuations in these factors, but also in the right-hand side, that pH diagram shows that in addition to these wide fluctuations, if you look at the rockshore habitat, which is where I work, pH can be kind of lower on average versus the kind of open ocean ambient pH that we expect, which is around like 8.2. And so many intertidal species, including some of the ones shown here, have become kind of these model systems for trying to understand stress physiology in general. And then also, we think we can apply what we learn in these species to predict responses to climate change and ocean acidification and maybe other marine species that inhabit the open ocean or other habitats besides the rocky intertidal. Just because currently they're facing really stressful conditions that we might predict to occur over the coming century with continued global change and ocean acidification. And additionally, with global change, we expect increased variability and increased prevalence of extreme conditions. And so perhaps we can learn something from how these species deal with variability today under current conditions, and maybe that can inform how other species and other habitats might deal with future changes in variability in abiotic conditions. And so the particular species that I'm going to tell you all about today, my study system for my dissertation was Tigriopus californicus, which is a harpacticoid copepod. And it's found all along the west coast of North America, distributed from Baja California, Mexico, all the way up to Alaska, as you can see in this diagram from my committee member, Suzanne. And they're small little guys, as you can see here on the right. I think this is a little video I'll try to play. Yeah. So they can form these really dense kind of monocultures in tide pools. So they are restricted to inhabiting just these rock pools in the highest part of the intertidal zone on the shore. And just to tell you a little bit about their ecology, to explain why they exist in this kind of very specific habitat. So you can see in this picture kind of different rock outcrops on the left and right where Tigriopus pools might exist. And they're separated by this sandy beach. And so what we know about Tigriopus from previous research is that there's low dispersal among isolated rock outcrops, even if they're only separated by less than a kilometer. And that's because Tigriopus splash pools are a refuge from predation. So if Tigriopus copepods happen to be washed out into lower pools on the shore, they're almost immediately snatched up by either anemones or tide pool sculpins or other predators. So they exist in these higher pools in the intertidal because that's where predators can't survive because conditions are more harsh, higher on the shore. So in general, there's pretty high movement of copepods among tide pools within a rock outcrop, but because of this predation, there's really low probability of individuals moving between rock outcrops. And so I'm going to start to transition now into part one of my talk, which is about responses to pH. So Tigriopus has been studied for decades. It's becoming this well-known model system for many things, including nuclear mitochondrial incompatibilities among hybrids of very differentiated populations. It's a good system for looking at ecotoxicology, or how they respond to various heavy metals or other chemicals in the water, just because again they have this very short generation time and are easily reared in the lab. So it's amenable to these kind of multi-generational studies. And it's been well studied for intertidal ecology. But what I'm most interested in is how it's been studied for its potential for acclimation and adaptation to climate change or other stressors. And so Tigriopus has been well studied by many labs for its responses to temperature, salinity, and oxygen, but we really don't know much about its responses to seawater pH. And so because Tigriopus lives at the upper edge of the intertidal zone, or even above the high tide line, it's living at the most extreme part of this marine habitat. And so we might expect that pH conditions in these pools would be even more extreme than pH conditions that have been measured historically in tide pools that are lower on the shore, that contain more diversity. So we don't really have a good understanding of pH dynamics in Tigriopus pools in the field, but I tried to characterize pH myself just to get a quick understanding of the extreme pH conditions that could happen. But again, I don't have high resolution sampling by any means. But what I did with my trusty beer bottle sampling method, which I took back to the lab at Friday Harbor, and you could do pH measurements in their analytical lab with a spectrophotometric method. I took samples at both dawn and dusk to try to capture the most extremes throughout the day. So, in the morning after everybody in the pool has been respiring all night, you would expect pH to be very low. And then at dusk, you would expect pH to be higher due to all the photosynthesis going on if there's any algae in the pool. And from this discrete sampling effort that I did across a couple of years, I found that pH in these pools can range from a little below 7 to well over 9. And maybe this doesn't seem like that big of a difference, but pH is on a log scale, and so a change of 2 units is very extreme. Whereas again, in the open ocean, pH is typically pretty stable around 8.1, 8.2. So I think, you know, it would be really important to understand how populations respond to this very dynamic, important abiotic factor in these pools. And so my research questions for this section is, how is the life history, including the fecundity, survivorship, and development of Tigriopus affected by pH? And then I wanted to get at, you know, this question of past history and current performance and maternal effects. So does the developmental pH exposure experienced by a mother influence her performance in new pH conditions or the performance of her offspring? And then I wanted to compare different populations to see if they have similar responses to pH over time, or perhaps, you know, there's some differentiation among populations in how they respond to pH. So again, as Bob said, where I've been working is at Friday Harbor Laboratories, where they have an ocean acidification lab where you can modify seawater pH, and also an analytical lab to characterize your seawater pH treatments. And the study sites that I focused on were kind of, you know, north, south, east, west sides of the island. So it includes Rubentart County Park, a site right near the labs, Cattle Point at the southern tip of the island, and Dead Man's Cove on the west side. And these sites are separated by, you know, like 15 to 30 kilometers, depending on, you know, how you draw your distance along the coast. But they're pretty close geographically, but distinct in terms of, you know, Tigriopus dispersal potential. And so here's my general design. I did 3 experiments from the F0 to F3 generation, and I'll walk you through this. And I'm going to tell you today about, this is what I did for experiments in 2017, but I did all of this twice pretty much. So I also, I repeated the experiment in 2016 and 2017, but I'm just going to show you results from 2017, but results across years were pretty similar. And so what I did was I took copepods that I collected from each of my field sites, and then I allocated them to these 3 different pH treatments. And so I chose 7 as my lowest treatment because that's around the lowest pH that I observed in the field. And I had to make pH 8 my highest because that's about as high of a pH as I can achieve in the experimental system at Friday Harbor. And so I allowed gravid females from the field to acclimate in the lab for a few days, put them in the pH treatments, and then they produced F1 offspring. And then they developed in those treatments. And then the second experiment started with taking gravid F1 females then, and then allowing them to produce F2 offspring in those same treatments. And then finally, my third experiment includes these reciprocal transplants. So that's when I'm taking gravid F2 females, and then I'm either keeping them in the same pH treatment that they were reared in or moving them into a new pH treatment. And then I track the development of F3 offspring within those treatments. And so as you can see, by experiment 2, I ended that middle pH treatment, and I'll tell you why in a little bit. And so my response variables throughout these experiments was fecundity, or nauplii, those are babies produced per female. And the way reproduction works in Tigriopus is that females will mate with a male, store his sperm, and use that to produce multiple clutches of eggs throughout our whole lifetime. So I just kind of measured all of the nauplii produced by multiple clutches over a 2-week period for everyone. And then I also, once I had nauplii, I tracked their survivorship and development over time by doing these weekly counts of all my copepods and how many copepods are in each life stage to be able to track, you know, who's developing into which stage over time. And just so you know what life stages exist for Tigriopus, they have many, but I kind of just divided it into nauplii, juveniles, and then adults, which you can identify by sex pretty easily just by morphological features. And so getting into some experiment 1 results first. Again, this is when I just took females from the field and then put them into these different pH treatments and tracked their offspring. Again, I had my 4 populations and 6 replicate cultures for each treatment and population combination for a total of 72 cultures that I was tracking over time. And so here I'm just showing you specific population comparisons, and I'll do that throughout the course of the talk. Here's just what happened at week 1 of the experiment. And the point of this slide is that, fecundity initially was kind of all over the place, which isn't surprising because they're, females that were reared in presumably different environmental conditions in the field. And then throughout a lot of my figures, I'll be showing you statistically significant differences among groups using these letters above the error bars. So if they have the same letters, they are statistically the same. And if they are different letters, they're different. But despite these kind of like variable population dynamics in the beginning of the experiment, now I'm going to show you what happened towards the end of the experiment at week 6. So here I'm showing you the effects of pH on total culture size at week 6, or the end of the experiment. And as you can see, they all kind of converged, all populations, where the largest populations or cultures were observed in pH 7, whereas cultures were smallest in the higher pH treatments. And so this was just due to high mortality in the higher pH treatments, and additionally, development was slowed in the higher pH treatments. Now I'll move on to experiment 2. This is where I'm taking gravid F1 females and then tracking their F2 offspring and their development over time. And so before I was kind of showing you slices in time throughout the experiment, now I'm trying to show you trends in culture sizes over time. So the x-axis has weeks. This experiment lasted a little over a month, and I'm showing you the number of F2 individuals normalized by the number of F1 mothers. And so a few things I want to show you. Populations are separated into these different facets, so these 4 different squares. And so F1 fecundity was highest in pH 8, or in females that were reared in pH 8. So as you can see, that for most of the populations, there's kind of this initial peak in population size, which is due to this like really rapid production of nauplii only in pH 8. And different pHs are indicated by different dashed lines. However, despite these kind of early booms of nauplii in pH 8, there was also very high mortality. So it was kind of this big peak and then a crash. Whereas in pH 7, where fecundity wasn't as high, survivorship was very high, so there was more stable culture dynamics over time. But then with pH 7.5, there was low fecundity combined with low survivorship and kind of these rapid culture declines. And so for logistical reasons, this is when I decided to end that middle pH treatment and just continue with the most extreme pHs. And I just wanted to point out here, we didn't see this developmental effect of pH 8 in Dead Man's Cove. And I want to point this out because across a lot of my experiments, it seemed that Dead Man's Cove tended to have different dynamics from the other populations. And so some conclusions from this experiment. For F1 fecundity, it was highest in pH 8, which is interesting because that was the most stressful pH treatment in experiment 1. But if we look at F2 survivorship, there are similar trends across different life stages. In general, survivorship was lowest in the highest pH treatments. Now, if we move on to experiment 3, again, where I employed these reciprocal transplants. So I'm going to focus in on cattle point just as an example, but trends were kind of similar across populations. What I'm showing you here on the x-axis are my pH treatment combinations, right? So the first number is where mothers were reared, and then the next number is where they were transplanted to. And then I'm showing you the number of nauplii produced per female. And here there was an effect of developmental pH, where mothers that were reared in pH 7 had higher fecundity than mothers reared in pH 8. And actually by this generation, so this third experiment, for some populations, the copepods that were being, had a history of rearing in pH 8 this whole time were really on their way out. So if I wanted to extend this experiment for another generation, I don't think it would have been possible for the higher pH treatment. And then if we look at now the F3 generation and how they did, here I'm showing you a proportion of mortality. So yeah, between the initial number of nauplii that were produced and then later on in the experiment, what proportion died. You can see that for the copepods that have been reared in the lowest pH throughout their whole, their parents were reared in pH 8, now they're reared, pH 7, and now they're reared in pH 7. Mortality was very low, but mortality was much higher in pH 8, which has been occurring across all of these experiments, right? But there is also this effect of mom's developmental pH experience. So if mom was reared in pH 7, her offspring did better than those with moms that were reared in pH 8. But if mom was reared in pH 8 and you're not doing so well, you do have the potential to have some improvements if you are moved into lower pH conditions. So it seems that this effect of pH is reversible. And this pattern held across populations. And so my conclusions for this final reciprocal transplant study, in general, cultures do well in low pH, cultures do poorly in stable high pH. But if you are in high pH and you are moved into lower pH, your performance can improve. And then, this goes the same way in the opposite direction. If you are in lower pH and move into higher pH, you will begin to decline. So, yeah, this was interesting. And again, I saw this across 2 years of repeated experiments. And back to my original questions. So what does pH do with life history and population dynamics? So as I said before, population growth is kind of optimal in lower pH. And so what do I think about this in context of what happens in the field? So in the field, these copepods might only face relatively short-term exposures to higher pH levels, whereas in this experiment, higher pH exposures were for extended periods. So perhaps in the field, they are able to withstand kind of more, short-term excursions into higher pH conditions for, a few hours, say when algae are photosynthesizing in the middle of the day. Alternatively, maybe, pH does get high in pools in the field all the time, and that leads to decline of copepod populations in the field. But as I mentioned earlier, these copepods exist in metapopulations on rocky outcrops, right? So different pools that are kind of connected by copepods moving around. And so potentially, if there's variability in abiotic conditions among pools, maybe because they're at different parts of the shore, different sizes, things like that, variability in space might underlie resilience of the entire metapopulation, right? So say some pools die out because they have high pH, maybe later they can be recolonized from other survivors in other pools, right? And so about this question of maternal effects, I found that fecundity of the F1 generation was highest after developmental exposure to pH 8, which was, the stressful pH condition. And this is kind of an interesting result that I've thought way too much about. And so you can, if you want the long story, you can ask me about it later. But in short, I have a few different hypotheses. Perhaps it's kind of like a reproductive bailout strategy that might be advantageous in these really dynamic environments, or I do have hypotheses related to kind of like density dependent population dynamics in these isolated pools, colonization ability, effects of population size on pH. So again, if you want to hear more, ask me about it later. And then, so again, these effects of pH are reversible. So there is potential to recover after exposure to, stressful pH conditions. So now to move on to part 2. So throughout all of my pH studies, I just noticed that these populations are different, right? So even though they all pretty much have the same responses to pH, other things are different. So they have different morphology. They have different colors that seem pretty consistent over the years. And they have different life history just in general across conditions. So, development speed, overall population sizes, things like that. So I wanted to quantify these differences and try to, in terms of morphology, and then also see, how genetically differentiated are they? And so my questions for this section are, do morphological differences among populations persist across years in the field? And then also, do generations of rearing in a common lab environment kind of erase these differences, which would suggest that they're environmentally determined? Or, do they persist, which would suggest that they're genetically determined? And then I also wanted to know, so, is there a genetic component to morphological differences, but is there also an environmental component? So does your developmental exposure to pH, temperature, and salinity affect your size as an adult, or, your morphological features? And then also, are there differences between the sexes in these effects of the environment? So I took many morphological measurements, and some more than what I'm showing you here, but the ones I'm going to focus on today are just head length, because I think it's a pretty good, proxy for overall just size. And so I'm just going to tell you a little vignette about the effects of developmental pH, and I'm not going to elaborate on the other work, but again, you can ask me about it later. So I characterized morphology of copepods at each of my study sites, just, collected them from the field and measured them. And I'm going to focus on results from 2017 for this part of the talk, but I did these measurements across 2016 to 2019 to see, again, if morphological differences among populations were consistent over time. And then I took these copepods, and then I produced F1 offspring, and then reared those offspring in different pH conditions. And additionally, I did this with salinity and temperature treatments. And then I measured adults that had been reared in these conditions since hatching. And so here's what I found. So again, this is just trends of, what size are copepods in the field. And I'm showing you females on the left and males on the right. Across years, so this pattern held across 4 years. Cattle point copepods are larger than the other populations, and dead man's is kind of an intermediate. And these are very small measurements, right? So it's hard to kind of feel for what difference does part of a millimeter make. But these differences were so pronounced that I was pretty much able to just look at a batch of copepods and know where they were from based on their size and appearance. That's how different they looked. So now for the effects of pH, the developmental pH on adult body size. So for females, I found no effect of pH. So what I'm showing you here are data from just within pH 8, but similar trends happen across the other pH treatments. And so what I'm showing you is the F0 generation with the black points, and then the F1 generation in teal points. And so you can see everyone kind of gets smaller after a generation of rearing in the laboratory, which I think is pretty common for Tigriopus, but these population differences are maintained. But if we move on to now F1 males, there was an effect of pH where higher pH led to smaller copepods. And so if you remember, higher pH was more stressful and led to, higher mortality, slowed development. And so additionally, in males only, body size was smaller. And also within each pH treatment, population differences were maintained. And this is interesting just because there's kind of a lot of research now that's showing that relative to males, females have superior tolerance to a variety of abiotic stressors, including temperature, salinity, exposures to various chemicals. This is a lot of work that's being done by my committee member, Suzanne. So this just seems like maybe another interesting little nugget of information that might contribute to that body of research. So back to my original questions. Basically, are morphological differences among populations genetically determined? It seems that they are, yes. And then, so for the effects of all these various abiotic factors on body size, I'm not going to show you the details, but I also found that rearing in lower temperatures, so like 10 degrees Celsius versus 20 degrees Celsius, led to larger body sizes, which is a commonly observed pattern for ectotherms. And then, but also interestingly, higher salinity, so 55 parts per thousand, relative to 32 parts per thousand, or like ambient seawater, led to larger body size. So that was an interesting result. And then additionally, are there differences between the sexes? Yes. So what I found that in males only, smaller body size occurred when they were reared in higher pH, but additionally I had this variable temperature treatment, and they had smaller body size in that variable temperature treatment relative to just like the average stable temperature of that variable treatment. And so, whereas, you know, for both of these treatments, females were unaffected, right? So it seems like males may be less tolerant to stressful conditions or changing conditions. And so now to move on to my genomics part. So again, as Bob said, this is a small little project I did in a NSF GRIP internship at the Northwest Fisheries Science Center. My mentor was Krista Nichols, who was really amazing and who took a lot of time to teach me, you know, library prep and bioinformatics. And so this is just a great learning experience to get these skills for population genomics. And so I used a pooled restriction site associated or RAD-seq approach to characterize the population structure of my study sites. And again, so I had to use a pooled approach just because it's challenging to get enough DNA out of an individual copepod for next generation sequencing methods. And RAD-seq is a method that uses restriction enzymes to kind of, you know, cut out a random subsample of the genome. So I'm only sequencing, you know, part of the genome versus the whole genome, which would be more expensive. And so my methods, I just collected hundreds of adult copepods from each of my study sites. And then I extracted DNA from 5 pools of 300 copepods from each of my sites. And then I have 5 pools because within that Dead Man's Cove site, I actually collected 2 distinct pools. I just wanted to test if, you know, this cove contains kind of 2 separate outcrops. One is higher on like higher pools, one has lower pools. I just wanted to make sure that what I was considering, you know, one population was indeed just one population. And then, so from these 5 pools, I prepared an SBF1 RAD library, and then I sequenced my library using a MySeq at the NOAA lab. And so for the bioinformatics methods, I demultiplexed my reads using stacks. And then I did my analysis using the pool party pipeline by Micheletti and Narum. And so this pipeline contains kind of these analysis modules. So the first module is this PP-align module that does, you know, quality trimming, PCR duplicate removal. Then it aligns my reads to the Tigriopus reference genome. And we have a very nice reference genome for Tigriopus, which is great. And then I called SNPs with BCF tools with quality filters. And then the next analysis module is this PP analyze module, where I can conduct among population analyses with population 2, which, you know, is just for pooled samples. And then so after removing positions with less than 50x coverage, I had 919 SNPs to work with for final pairwise analyses. So definitely a modest number, but I think sufficient for just my simple question of how genetically differentiated are they. And so here as my first result, I'm showing you below the diagonal on this chart is my average pairwise FST values. And so FST is just a measure of population subdivision, right? So 0 means no subdivision, 1 means total subdivision. And then above the diagonal, I have approximate shortest distances between the sites along the coast. In kilometers. So I'll highlight some parts of this table I want you to focus on. So, you know, the lowest FST value that I had was between, you know, those 2 pools within Dead Man's Cove. So it seems that it's appropriate for me to, treat Dead Man's Cove and these separate outcrops, as one population, because there's relatively much lower subdivision. And then the highest differences occurred between the east side and west side populations, right? So between Dead Man's on the west side and Friday Harbor and Rubentart on the east side. And then I'll just highlight, Cattle Point seem to be kind of equally differentiated from all other sites. And just to show you another way of looking at these data. So here's a little PCA with my 5 pools. So Dead Man's Cove is differentiated from everyone else, but Cattle Point is kind of like this intermediate between them. And so I think that maybe I might have an explanation for this based on currents, which I'll tell you about. So just to remind you kind of where San Juan Island is in the grand scheme of things. So it's kind of, right above Olympic National Park. You can see Seattle in the bottom right corner. So the west side of San Juan Island, which I have circled there, is exposed to, incoming swell from the Pacific Ocean, whereas the east side is kind of protected facing the San Juan Channel. So the west side and the east side are kind of known to be just ecologically distinct just because of these really different geographic and oceanographic conditions on either side. And then I'm going to show you just some screenshots from the Pacific Northwest Current Atlas to try to show you kind of how currents work here. So now I'll just overlay just, in general, what the PCA shows. And so you can see Dead Man's is that green point on the west side of the island. And the way the currents work around San Juan Island, is basically they move from north to south and south to north as, the tide comes in and out. So you can see as they're kind of moving from south to north, there's kind of these strong currents that rip up the west side, but the currents kind of diverge at Cattle Point. So I feel like there's a probably equal probability of copepods being carried onto either side, from Cattle Point in the south to either the west and east side. So that might kind of explain why Cattle Point is like pretty similarly differentiated from both the east and west side. But then if you look at the currents kind of going in the opposite direction now, the tide has changed. I think just there's less of a chance for movement of individuals across the northern end of San Juan Island just because of how the currents move, but also, the shoreline is more convoluted up there. There's a lot of embayments. So I think there's less, lower probability of migration, between Rubentart across the shortest distance to Dead Man's Cove. Just gonna take a quick sip of water. Just in general, I think there's potential for maybe like an isolation by distance interaction with currents explanation going on here. So moving on to my final part about local adaptation. So in general, after all of this, I'm pretty certain that there's at least, some genetic subdivision between my populations, of course, but I also want to know, is this differentiation influenced by adaptation to local conditions, or is it more of an effect of just, they're isolated, there's not a lot of exchange of individuals, and genetic drift is happening? And so just to review, local adaptation is a pattern such that resident genotypes, or locals, have on average higher relative fitness in their local habitat than genotypes or individuals coming in from elsewhere. And so this process may be hindered by gene flow. And so, Tigriopus doesn't have very high gene flow between populations. So maybe it's a candidate for local adaptation. It's confounded by genetic drift. We know genetic drift can be pretty high in Tigriopus populations. There's a lot of population bottlenecks, so maybe it's confounded. It can be constrained by a lack of genetic variation or genetic architecture. And it can also be opposed by natural selection due to temporal environmental variability, right? And so we know that Tigriopus faces this temporal environmental variability, so maybe local adaptation would be opposed. And so as I've been saying, coastal systems are highly heterogeneous, right, which makes them a cool place to do ecology. And again, as I just said, this heterogeneity can occur in time, but also it can occur in space. And so that's what I was interested in for this chapter. You know, are there differences in space in the amount of temporal heterogeneity, right? So maybe some sites have more stable dynamics, some sites are more extreme and variable, and maybe there's local adaptation to those different levels of environmental variability. So thinking more on this like microhabitat scale versus larger range-wide scales. So local adaptation has been found for Tigriopus californicus, but more again on this really large latitudinal Klein scale. So unsurprisingly, southern populations, so like in California, can withstand higher temperatures and higher salinities, and northern populations can withstand lower salinities. But again, I was focused more on this, you know, very local scale, and interested in the effects of, again, differences in environmental variability versus mean conditions. So again, you know, does this heterogeneity hinder or, you know, help local adaptation? And so for this part, I only focused on 3 of my study sites. My questions for this part are, you know, how do the combined stressors of temperature variability and hypersalinity impact Tigriopus population dynamics? So many studies have looked at the effects of each of these factors in isolation. Fewer have looked at the interactions. And then I want to know, again, are any observed differences in, you know, responses to these stressors? You know, do they differ among Tigriopus populations, and is that due to genetics? And then, you know, finally, are they locally adapted? And so I use this 2-part approach. The ideal test of local adaptation would have been to do reciprocal transplants of individuals in the field. But that's not really feasible for this species. So what I did was I characterized environmental conditions within pools in the field to see if are there potentially different selective pressures among sites. And then I did a lab experiment where I established common garden cultures, and then I exposed them to factorial combinations of different temperatures with different levels of variability and hypersalinity. And these treatments were based on my field data. And so to get into my field results first. So what I did was I just went out to my sites, measured salinity in multiple pools every 2 weeks. I installed these temperature loggers so I can get pretty continuous temperature data. I did this across 6 months in 2018. And then I also used this kind of laser level equipment to measure heights of pools on the shore, because again, that's kind of a proxy for how extreme conditions might be in the pools. So higher pools are more isolated from, you know, incoming high tides. And so first, I'm showing you some salinity dynamics over time. And so, you know, I have date of survey on the x-axis, and I'm showing you different populations and their salinity dynamics in different colored lines. And so each point represents the mean of multiple pool measurements at each survey. And so I guess first thing is that salinity is crazy variable in Tigriopus pools. So it can be very low, almost fresh, and then can go to, I've had measurements of, you know, over 100 parts per thousand with copepods actively swimming in that water. So salinity is insane. But what kind of stands out to me from this graph is looking at the Friday Harbor population, where you can see at multiple time points, there is no variability among pools, and salinity is right around 32, which is about ambient seawater, which tells me that those pools were all probably flushed out at high tide, whereas at that same time point, at different sites, pools were not flushed out. So in general, salinity is lower and less variable at Friday Harbor, because it seems that they get flushed out at high tide more often. And so this is driven, I think, due to just local geology. So this is a picture of the shore near Friday Harbor. As you can see, there's kind of this narrow strip of the rocky shoreline. between, you know, the forest and the water. And there's just a really narrow range at which Tigriopus pools occur, and they happen to occur lower on the shore. So they're flushed out at high tide more frequently. Whereas at the other sites like Cattle Point, you can see there's this wider rock bench and a wider range of shore heights at which pools occur. So some pools maybe at the lowest heights get washed out sometimes, but then there are also way more extreme pools higher on the shore where you find Tigriopus. And now to get into some temperature trends over time. So again, I'm showing you trends across my 6-month study season. And I'm showing you on the y-axis daily temperature range. So the difference between, you know, the minimum and maximum that occurred within a 24-hour period. And each point, it represents one pool measurement per day. So as you can see in the summer, daily temperature ranges can be pretty extreme, so over 20 degrees within a day. But then as you move into the fall, air temperature becomes more similar to water temperature, and you see the ranges decrease. And then, so different points represent different populations. There's a lot of overlap. But if you kind of focus on the top edge of the cloud of points and the bottom edge of the cloud of points, there is this bit of, you know, differentiation where Dead Man's Cove, which is those darker points, has slightly higher average daily ranges versus the other sites. So, you know, slightly lower daily minimum, slightly higher daily maximums. The average daily range is larger by, you know, a little over a degree. So this is a pretty subtle difference, but it's interesting. And so, you know, based on these field data, what might my expectations be for my experiment in the laboratory? So I would expect that maybe Cattle Point and Dead Man's copepods would have higher performance in hypersaline conditions than Friday Harbor copepods, just because they have a higher chance of experiencing hypersaline conditions in the field. And then perhaps copepods at Dead Man's Cove would have higher performance in more variable temperature regimes than the other sites, just because they tend to experience larger daily temperature ranges than the other sites. So to move on to my lab experiment, here's the overall design. So I established common garden laboratory cultures for 3 of my populations. So they were kept in common garden conditions for 2 generations before the start of the experiment, which is hopefully reducing any environmental effects from when they were in the field. And then I had 7 temperature treatments, 5 of which were stable and 2 were variable. So there was kind of a variable low amplitude treatment and a variable high amplitude treatment. So they both had an average of 20 degrees, but, you know, different minimums and maximums. And then I had 2 salinity treatments, 32, which is about ambient seawater, and then 55, which is hypersaline. But, you know, it's not too crazy of a salinity for Tigriopus. It's pretty commonly experienced by them in the field. And so my response is kind of similar to what I did for all the pH stuff. I was, you know, counting copepods forever to track population dynamics over time. So I maintained kind of these mass static jar cultures for 2 generations. And at the end of it all, I quantified, you know, total culture sizes. But throughout the generations, I also took subsets of individuals and tracked individual clutches over time to get at F1 fecundity and then survivorship and development. And so the first results to get out of the way is there was high mortality in that highest stable temperature treatment. So I had to end this treatment early, and so it's going to be omitted from the rest of the results. But interestingly, I counted everyone when I ended that treatment. And it seems that when this higher temperature is combined with high salinity, there are more survivors. Again, so these cultures were all on the way out, but you know, I guess perhaps being able to hold on a little bit longer in this combination of high salinity and high temperature is advantageous in these high shore pools that are kind of roasting in the sun and evaporating all day. And this effect has been found by other investigators as well. And so now I'm going to show you some, you know, population dynamics. So here I'm showing you results for Friday Harbor, where I found a significant temperature and salinity interaction. So different salinities are shown in the different facets, right? So I have 32 on the left, 55 on the right. I'm showing you the number of offspring produced per female and those individually tracked clutches. And what else do I want to tell you? Yeah, so in general, fecundity was highest in the lowest temperatures, which is, not surprising, but it was interesting that fecundity was much lower for, even room temperature. And then once you got to like 25 degrees and that variable high treatment, there was very low production of offspring. So they seem to be the most stressful treatments. And so now I'm going to kind of overlay cattle point to do a population comparison. So I'm keeping the same graph, but I'm putting cattle point on top in blue. And so dynamics were pretty similar, except it seems that cattle point tended to have higher fecundity and that higher salinity treatment, but this wasn't true across all temperatures, which was interesting. And then additionally, I just wanted to point out, you'll see that a point is missing for cattle point in blue for that combination of 55 parts per thousand salinity and 10 degrees, just because development was extremely delayed in that treatment combination. And this occurred for all populations, but development was most delayed for cattle point in 55 parts per thousand, even across temperature treatments. There was a big population difference there. And so now I'm showing you survivorship trends over time. So I'm showing you, I was able to track again individual clutches across a month. And here I'm showing you like the proportion at each week that were alive out of the original number of babies in the clutch. So in addition to high fecundity in low temperatures, there's very high survivorship in low temperatures. But once you get to 25 degrees and above, mortality is very high. And again, within 55 parts per thousand on the right, we're missing the most extreme temperature treatments just because development was so delayed. So I couldn't track survivorship over time, just because F1 moms never developed in time to give me F2 offspring to track. And so in general, I was only able to show you, a few results from this experiment today, but here are some general conclusions that I got from these experiments. So as I said, fecundity and survivorship were highest in the lowest temperatures. And for these northern populations in Washington, actually being reared at like room temperature of 18, 20 degrees could lead to lower culture sizes. It's kind of stressful. And then what I want to stress is that culture growth was equivalent between that stable 20 degrees and that variable treatment that went back and forth between 15 and 25, but has an average of 20 degrees. So it seems that it's just, whatever your mean temperature exposure is matters more than the amount of variability. And then, if you think about that variable high amplitude treatment that goes back and forth between 10 and 30 degrees, those populations or those cultures did not so well, and it was not equivalent to stable 20 degrees, but I don't think it's an effect of the larger amplitude of variability. I think it's just an effect of any exposure to 30 degrees is stressful for these Washington populations. Even though temperatures in their pools can be, up to, above 30 degrees, but only for brief exposures in the field. And so development rate increased with temperature, which is, expected, but after about 25 degrees, that's when things kind of broke down and development was disrupted. And for fecundity and development, there were these interesting population differences where for both Cattle Point and Friday Harbor, I kept seeing salinity and temperature interactions, but for Dead Man's Cove, there was frequently only an effect of temperature and no salinity effect. So back to my original questions. Are observed differences in life history traits and stress responses due to genetic differentiation? Yes, that's what I've kind of been finding throughout all these experiments. But for the next question, the answer is maybe not so clear. And so I'll go back to my original expectations and walk through them. So this first expectation, that Cattle Point and Dead Man's copepods would have higher performance in hypersaline conditions than Friday Harbor copepods. Did I find evidence for this? Perhaps for that fecundity response and for Cattle Point only, where Dead Man's Cove had pretty similar dynamics to Friday Harbor in that higher salinity. And then for this next expectation, I really didn't find any differences among populations and their responses to the variable treatments. But then again, I don't think that I really found strong differences in temperature variability in the field. So in general, I don't think that's too surprising that I didn't find much evidence for that expectation. But in general, throughout this work, so, it's hard to really say how much adaptation is influencing Tigriopus evolution on San Juan Island versus, drift. Just because I still don't think, even with all of my field data that I collected, I don't think we have a really strong understanding of the selective pressures that they face. Again, you have to think more on like this microhabitat scale. And then, not only what happens in pools, but how do these copepods move among pools, and conditions among pools can be very different. So we really don't have a great understanding of abiotic conditions in these pools, just because, not many things live in these pools besides Tigriopus. We have more abiotic data for, lower parts of the shore. But I also, found other interesting results that might be consistent with local adaptation. So for the Friday Harbor population, so because those pools are lower on the shore, they are less likely to experience, extreme salinity or extreme temperatures. However, because they're lower on the shore, that also means that they are more likely to be washed out of pools and eaten by predators. And across all these experiments, I found that Friday Harbor, that population, even after common garden rearing, has faster development than the other populations, smaller population sizes. And so it seems that maybe there's selection going on there where, because there's this high probability of being swept out to sea and getting eaten, there might be this kind of life history evolution of, live fast, reproduce young. That might be a more advantageous life history strategy. So for some general conclusions, I hope that wasn't way too long. So it's rare for marine invertebrates to show higher fitness in pH 7 than under pH 8, which is like ambient open ocean, right? So these results are a new contribution to the body of literature that's documenting taxonomic variation in responses to changing carbonate chemistry in the sea. And this body of literature is growing rapidly because people want to know, how is ocean acidification going to affect everything. So I think it would be interesting moving forward to try to explore, the physiological mechanisms of acclimation to pH conditions in Tigriopus, much like we study, the CO2 vents near Ischia, Italy, where, things are naturally acidified. Maybe habitats such as, these tide pools would be a similar kind of, field experiment that you could do to look at, the mechanisms of dealing with low pH and adaptation to low pH conditions. So maybe some, gene expression work across different pH conditions would be pretty cool. And so although Tigriopus lives in a highly dynamic environment, it may not have a generalist physiological response to all abiotic factors that, it might regularly experience in the field. So they might not be able to withstand, every temperature that they might experience or every pH they might experience. But again, as I mentioned earlier, because they have these metapopulation dynamics and there's variability in space in different subpopulations, that might facilitate, whole population persistence because Tigriopus has such a rapid colonization ability. And then finally, while maybe I found some evidence for local adaptation and, specific traits to these microhabitat conditions around San Juan Island, it seems that here maybe most population differentiation is due to just isolation and drift. Even though the populations that I studied here are not, very highly differentiated by Tigriopus standards. And so just to move into some acknowledgments for funding, I've gotten help from many different organizations throughout all these experiments. It kind of takes needed a lot of support to be able to travel to Washington in the summer and, be able to stay in the OA lab and do all this work. So I really appreciate the support from all my funding sources. So now to move on to my people acknowledgements. So first I wanted to thank Bob, my advisor. I just chose this picture that I found in the E&E website just because we both look very happy, and I think it really represents Bob well, just because he's one of the kindest people I've ever met, and his kindness has really helped pull me through some really tough times throughout my PhD. And so when I've been in the depths of despair, Bob's positivity and support has really helped get me through. Ever since he came to Stony Brook, he's been just a wonderful committee member and advisor, and I really appreciate all that you've done for me, Bob. So now to move on, many people to thank. So my dissertation committee, so people from Stony Brook, so Stephen and Josh, besides just being great committee members and advising me on my science, I just wanted to point out how grateful I am, and I think all the graduate students are, to you both for being really involved as, graduate program coordinators, just in improving graduate students' lives and well-being. And you're just really committed to helping us all and being supportive to all of us. And so I really appreciate that from both of you. And Suzanne, my external committee member, who is, Tigriopus californicus extraordinaire. Your research has shaped my dissertation before I even asked you to be on my committee, and your really careful review and comments on my manuscripts has made my dissertation just infinitely better. So I really appreciate the time and care that you put into, helping me, even though you've never even, met me in person. So I really appreciate that. And then I also wanted to thank my former advisor, Diana Padilla. So throughout my PhD, your guidance and support and willingness to discuss science and your enthusiasm for science has really shaped who I am as a scientist. And you really helped turn me, into someone who can do science independently versus, who I was when I came into Stony Brook fresh out of undergrad. And you also introduced me to Friday Harbor Labs and the whole community, and that changed my life. So I can't thank you enough for that. Just in general, my experience in your lab has really shaped who I'm going to be as a scientist and a mentor and a collaborator moving forward from Stony Brook. And so other people at E&E I would like to thank. So Donna, Martha, Melissa, and Fumio, and Irene. You all make the department run. Just knowing that you're there to support me has helped me in so many times, whether, any issue with getting reimbursed. I know Donna's gonna fight for me and get me my money, or, Martha is there for support to help me with any paperwork, give me chocolate whenever I want, or just Melissa has been a wealth of knowledge and has kept me on top of every administrative thing, and has also been there for personal support in many tough times. So I can't thank you enough, Melissa. And then Irene has been wonderful to just say hi to in the morning. And again, she just keeps the department running. So I'm really appreciative. And then kind of going back in time a little bit, I'm really grateful to my mentors at Dowling College, Rich, Vishal, and Chris. They were the first people to tell me that getting a PhD was even possible. And then besides just, putting that seed in my head, they did everything they could to support me and prepare me for getting a PhD straight out of undergrad. And, I wouldn't be at Stony Brook if it wasn't for them. So I'm very appreciative of all that they've done for me. And then I just have these little icons for AMS and SICB, just because through those societies, they've given me a lot of financial support. as well. They just formed these professional networks that has been really great for me as a up and coming scientist. So they're really great organizations. I'm very appreciative. And then within NOAA, I have a lot of people to thank. So within the Northwest Fisheries Science Center in Seattle, where I did my internship, Krista was an amazing mentor, just so generous with her time and resources, and basically just allowed me to do whatever the heck I wanted, including, you know, analyzing samples for my dissertation. So she was awesome. And then I also wanted to thank Abigail Wells and Piper Schwenke for they were basically like there whenever I needed help with anything in the lab and I had no clue. So they told me where everything was, how to use everything. Basically just, I wouldn't have made any rad libraries if it wasn't for them. And then Giles was, you know, the bioinformatics extraordinaire, who again put up with me knowing nothing and taught me a lot. And Shalyn Bush was also a great person to chat with for just talking about ocean acidification and research in general. And then outside of my dissertation, I also did work on mussels and ocean acidification at the Northeast Fisheries Science Center in Milford, Connecticut. And Lisa Milkey there has just been a wonderful source of support and a wonderful mentor, and I'm so grateful to Lisa. And Shannon was our kind of ocean acidification chemist extraordinaire, and she taught me a lot about doing OA research. And then Dave, Dylan, and Mark were just really great at you know, teaching me everything related to bivalve aquaculture, rearing microalgae. They're just like super experts in everything related to aquaculture, and being able to work alongside them was an amazing experience for me. And so just in general, these are the people that I worked with the most at NOAA and spent the most time with, but almost every person I've ever interacted with at a NOAA lab has been amazingly kind and helpful. So I can't speak highly enough of those labs. So now to move on to my buds. Grateful to my cohort. We've had a lot of great times. And so you've kept me going throughout the many stresses of grad school. In addition to just the fun, you're all, you know, geniuses, and you've taught me so much, whether it's, you know, how to get through biometry, how to code. Just having you all as resources has really, you know, taught me so much. And in general, at Stony Brook, there's been many clubs and organizations that have kept me going, including GWISE, which has been such a great way to meet people from other departments. I love all of our birding outings. Thanks, Doug, for teaching us your expertise. So in general, the grad student community has just been amazing. And so this is just to represent everybody in the department, just because I think within E&E and also in SOMAS, I've approached probably every professor asking a question or asking to borrow something, and you've all been very kind and generous with your time and resources, and I've learned so much from you all, and, you know, students as well. Besides just being fun to hang out with, you've all taught me so much through Tuesday Noon or just random meetings where I ask you about what you're an expert in. So it's been an excellent community. Then I wanted to focus on my lab mates, both past and present. And then even more so, I wanted to focus on Maria, David, Allison, and Jose, who I think I've spent the most time with out of all my lab mates. We've been through some tough times together, and your support has really helped me get through those tough times. I can't thank you enough. And now getting into Friday Harbor, I need like a credit reel to thank all of the amazing people at Friday Harbor. From when I first came to Friday Harbor in 2015 all the way to, you know, 2019, I've met so many amazing people. It's just this community of just truly amazing people, and there are times where I couldn't even believe that I was a part of such a community. So many people that I could just go up to and ask any scientific question, people I could just borrow equipment from. You know, it's just this great community to work in. And as I said, being at Friday Harbor changed my life. And so within all of these people, I just wanted to focus in on, you know, my dorm Q crew. So people that I've lived with, my great graduate student friends. I think all of these people have either, let me sleep on their couches, or take their car to go to a field site, or have cooked me meals, or, brought me chocolate when I'm dying in the lab counting copepods, or, fixing me a strong gin and tonic after a long day in the lab, right? So it was just really my second family out there in Washington. And I can't thank you all enough for just being so fun and supporting me in every way possible out in Washington. I just wanted to do a quick extra shout out to my good friend Sasha, who's in the bottom right corner there. Besides just helping me with my experiments all the time, there were many times where I was in the depths of, experiments that would never end. And I was so close to, quitting in despair. But you were there for me, and you pulled me back from the edge. And I can't thank you enough for your friendship. All right. And then, just 1 second. All right, it'll be over soon. So this is like my home squad. So on the left there is my good friend Robert. And then, so he's been a good friend of mine since junior high. He accompanied me on my first ever trip to the West Coast back in 2015 for the FHL OA course. There we are, hanging out in Seattle. He's just been a consistent, great friend always, who has always supported me. And even if we go a long time without chatting, every time we re-meet, it's like, no time has passed. So thank you, Robert. And in the middle there are my two best friends, Tori and Jenny. Again, kind of like a lot of my home friends, I'm sure they don't really understand what the heck I'm doing with my time, but they totally support me in every way possible through, the ups and downs of life, both professional and personal. And they've just been, I can't believe how lucky I am to have them in my life, even when we're all on different sides of the country. We've been there for each other again since, running together in junior high. And then on the right there is my brother and sister-in-law. We always support each other. We're siblings, and I can't thank you enough for supporting me throughout my PhD. And I know you're going to continue to support me, in everything I do after my PhD. And finally, to end, I just wanted to thank my parents for, supporting me unconditionally through everything I do. I know that my life choices may stress you out at times, especially, not knowing what this degree is going to end up in terms of a career. I know I've gone kind of the non-traditional route, but you've been open-minded and totally supportive of everything I do. And, even visiting me out in Washington, here we are at Cattle Point. And, you're the hardest working people I know. And I know that you work so hard so that you could build a life for me and Matt, so that, all we had to focus on was achievement, right? So the only way I've been able to get to, a PhD and to graduate and then now move on to a postdoc is because you've created this life for me where I can just throw my whole self into my academics and being the best I can be. So without your support, there's just no shot that this would have been possible. And in general, I know this sounds like corny, but if and when I get this PhD today, I really see this as our achievement, and that it belongs to us. It's not just mine. So that's it. If anybody has any questions, I'd be happy to take them. So yeah, you can just raise your hand in the chat, actually in the participants screen. Oops, I'll leave the conclusions. Ooh, all right. I think I saw that Sasha is raising her hand. Let me do the gallery. Can you unmute yourself? Okay, cool.
Hey, Alyssa, great talk. It's really nice to see this all come together. So I'm wondering about and we've talked about this a lot, like why they do well in pH 7. And I'm seeing your results from the temperature and salinity stuff. I'm wondering how you think that could inform your interpretation of that. Because it seems like that you said at like at 20 C, they didn't do as their fecundity was lower and survivorship was lower in the lab. And I think, was that your temperature?
Yep.
In the OA stuff? So I'm wondering if there's like, you think there's maybe some interaction there, and what the interpretation might be.
Yeah, for sure. Yeah, so I did the pH work before the temperature and salinity work, and I chose 20 degrees just because I was like, I need to get, multiple generations. I have a limited field season, so I tried to kind of push the limit of temperature to get them to develop faster. So there's kind of a trade-off there. But certainly now that I know better, if I had reared them at lower temperatures in combination with my pH treatments, perhaps overall, I would have gotten, higher culture sizes. But yeah, there could be, instead of just culture sizes boosting across the board, some interaction with temperature stress and pH stress. And that's what's tricky too, right? Like I looked at the interactions between temperature and salinity because those are relatively easy factors to manipulate together. But when trying to manipulate temperature and salinity and pH together, it's like not fun. So we know that salinity influences pH, right? Because salinity affects buffering capacity of the seawater, temperature affects pH. So I would have to think more about how all of these things may be changing together in the field and then how they might interact in terms of culture dynamics. But that's a great point that I definitely have not addressed here and would be tough to do, but is possible. So thanks.
Thanks. Yeah, complicated system, right? Lots of things to think about there. Awesome work.
Thanks. All right. Maybe I'll look in the chat. Scrolling for hands. All right. Any other questions before we wrap this up? Okay, I don't think I see anything. So.
Going once, going twice. Okay, so I think it's time to adjourn for the committee to meet and have cocktails. That's because that's what really happens behind closed doors. And to ask Alyssa some questions about her research. I want to thank everybody for coming, and yeah, so I guess we're going to stay on, but we'll ask everybody else to leave the call, or we're going to kick you off.
Get out.
Okay.
Thank you everyone for coming. I really appreciate it.
Okay, I'm also going to stop the recording, right? Unless y'all want to also record everything else.